Mic on? Oh, there it is. We saved the best for last. Thank you for staying to hear this symposium. This is a symposium that's sponsored by the Pancreatic Cancer Action Network in conjunction with APA so that we can highlight the work of young investigators who are supported by the network. For those of you who don't know me, I'm Margaret Tempero. I'm a medical oncologist. I am director of the Pancreas Center at UCSF. I'm also chair of the Scientific and Medical Advisory Board for PanCAN. Excuse me. Joe, you want to introduce yourself?
Hi, my name, here we go. Thank you. Joe Hines, I'm from UCLA and we're looking forward to this session.
So with that, let us invite the first speaker up. Nutrient availability in the microenvironment rewires pancreatic cancer cell metabolism. Alex Muir, University of Chicago.
Awesome. Thank you very much for the introduction. And thank you very much to the APA and PanCAN for the opportunity to share our group's work. As Dr. Agul alluded to a few talks ago, there's a lot of interest in the pancreatic cancer field to understand the cellular metabolism that fuels pathological phenotypes in pancreatic cancer. That is, what nutrients do pancreatic cancer cells need, and what metabolic processes do they require to process those nutrients to fuel downstream pathological phenotypes like immunosuppression, therapy resistance, and unchecked growth. And we're really interested in this, not just from a basic biochemical perspective, but also from a translational perspective as well, because these are places where we can readily intervene therapeutically. If we know what nutrients pancreatic cancer cells require, we can intervene via interventions like diet to starve the cells of those nutrients. And this is something that increasingly patients are looking for advice on as well. And then all of the metabolic enzymes and transporters that process these nutrients, they all actively handle small molecules all the time, so they're ligandable. So it's likely we'd be able to find a drug for a metabolic pathway if we could just know what the critical metabolic pathways are. So what are the challenges in finding these metabolic pathways? And so what I want to highlight is really a mismatch between what our model systems can give us and what our metabolic analytical technologies allow us to do. And so most of metabolomics technology requires a set of pure cells because we really don't have spatial or single cell metabolomics technologies yet. And so the best models for doing metabolomics experiments are cell culture based models where we can get a large amount of purified cellular material. The problem is these models aren't really physiological. And a big part of this is because these models are kind of like the cellular equivalent of a Vegas buffet. There's all of the nutrients that these cancer cells would want in vast excess. And I don't know about you, but my nutrition and metabolism looks different in Vegas than it does at home. What we really want to be studying is actually these cancer cells in situ when they're faced with poor perfusion, limited access to nutrients, and competition for those nutrients from other cells in the microenvironment. And this really looks like a much less enticing buffet than what we're giving the cells in standard culture models. Unfortunately, we just don't really have the analytical capabilities to look with precision and accuracy at cancer cells in situ yet. And so what our group wanted to do was find a way to get past this roadblock. And we thought a good way to start to get past this would be to understand what the nutrient microenvironment of an actual PDAC tumor looks like. What does the buffet actually look like in vivo? And so we first started by asking how are pancreatic tumors fed. And so pancreatic tumors, much like normal tissues, are fed by afferent blood vessels that come in. They break out into a capillary network, which acts as a filter. And this filters out a nutrient-rich fluid called interstitial fluid. And this is actually what brings nutrients directly to the cancer cells before becoming the lymph. And so we reasoned if we wanted to know what was locally available in the microenvironment of PDAC tumors, we'd need to take a look at the tumor interstitial fluid, or TIF, coming from these PDACs. And so briefly, that's what we did. We adapted and developed some techniques to isolate interstitial fluid from KPC PDAC mouse models, and then developed an analytical pipeline to go in and quantify the nutrient levels, their absolute concentrations for hundreds of different nutrients in these biofluid samples. And so to sum up, going to map a metabolism here. For each one of the metabolites that are represented with the black dot, these are a nutrient now where we can go in and measure the absolute concentration of this nutrient or vitamin directly in the tumor microenvironment. This covers a lot of the circulating nutrients and vitamins, with the notable exception on the left of lipids, although we're currently working on the technologies to make those measurements as well. And I do want to say all of this data is now published and available, and we're working on developing additional analytical technologies to measure other things like cytokines, clinical chemistry, and electrolytes. And so hopefully this will be a nice resource for the PDAC community to have kind of a high precision measurement of what's going on physiologically in the tumor microenvironment. All right, so now that we have these measurements, our group really wanted to ask, what can we learn that's new about how tumors metabolically function from having this kind of atlas or menu of what's available in the tumor microenvironment? And this is work from a really awesome graduate student, Juan Apiz, in our lab. And he got a lot of support from a technician, former technician in our group, Lindsay Desire-Zinsky. And so what Juan and Lindsay did was they decided to build a new cell culture model, which we call tumor interstitial fluid medium, or TIFM. And so they took the nutrient concentrations that we got from our metabolomics pipeline, and we were able to develop a cell culture model where we have 115 of those metabolites and vitamins at the exact concentrations that we measure in the tumor microenvironment. And we're really excited because this gives us a system now where we can ask how PDAC cells adapt and function when they're confronted with that nutrient milieu. And hopefully this gives us a nice marriage of the tractability of an ex vivo model system where we can do lots of mechanistic experiments while combining the physiology of the tumor microenvironment. All right, so Juan and Lindsay didn't want to stop there and just have a system that felt more physiological. They wanted to go a little bit more in depth and actually ask, can we prove to ourselves, demonstrate that the cells are behaving more in vivo-like in this system? And so what Juan and Lindsay did was they took KPC cells and cultured them in standard conditions, so the typical model, our TIFA model, or orthotopically transplanted them into a mouse and allowed a tumor to form. They then re-isolated the cells by flow cytometry and then performed RNA-seq on the cells. So we could use the transcriptional output of these cells in these different conditions as a proxy for their cellular state or phenotype, and then ask how, which of our models are better recapitulating what the cells are actually doing in a real tumor in vivo. And so there's a lot of ways to represent this data. I'll just show this one simple Venn diagram. So these are significantly enriched, curated gene sets from MSigDB. So these are coherent sets of genes that represent different cellular processes or pathways. And what I hope you can appreciate is that if you look at the pathways that are differentially regulated between a standard culture PDAC cell and cells that are growing orthotopically in a mouse, there's 1,200 differently expressed gene sets. And so even though these are the exact same cells, they're basically different beasts between standard culture model and what they're actually doing in vivo. TIFM to RPMI, there's about 900 differentially expressed genes, and we're really excited to see that there's this large overlap between the TIFM model and what's going on actually in the tumor. You can see the odds of this overlap happening by chance are quite low. And so we're really excited that we have this model system now where the cells are behaving more in vivo-like, and we can start to use this model to pick apart how these 450 cellular processes may be playing a role in pancreatic cancer cell biology that we just couldn't do before in our standard culture systems. Outside of transcriptional behavior, we also have additional evidence that these cells are behaving more as cancer cells actually behave in vivo. And so the KPC model recapitulates quite nicely the primary chemoresistance that's seen in human PDAC patients. Traditional standard culture models, however, PDAC cells are quite responsive to these drugs. What we found was in our TIFM cultured cells, they actually exhibit a fair amount of chemoresistance to the agents that are actually not working well in KPC models as well. So we think in addition to the transcriptional behavior being closer to in vivo, they exhibit chemotherapy responses that are more similar to how KPC mice actually behave as well. All right, so we have a model where we can now grow cells with tumor nutrition. It's more physiological, and the cells do appear to behave more like they would behave in a real tumor. So how can we leverage this to find novel metabolic adaptations? And so Juan went back to RNA-seq data, looked for metabolic pathways that are up in our TIFA model, up in the tumor, but not expressed in our PMI. there's a whole slew of these metabolic pathways. The one that Juan focused on is this enzyme ASS1 or argininosuccinate synthase. I'm showing the protein conformation of the RNA data here. You can see that this enzyme is heavily expressed in both murine and human PDAC, and our TIFM cultured cells recapitulate this expression quite nicely. In contrast, RPMI cells don't express this protein at all. So what is this enzyme doing in PDAC, specifically for cells in the microenvironment? And so ASS1 is part of the de novo arginine synthesis pathway, where cells can take aspartate and citrulline and make arginine. The other pathway that cells have to get this amino acid is by uptake from the environment. And so one clue that we had for what this pathway might be doing in PDAC was going back and looking at our interstitial fluid data, where arginine is one of the most depleted nutrients in the microenvironment, only present at about 2 to 5 micromolar. And so perhaps in PDAC, this uptake from the environment pathway is shut down, and this pathway might become quite indispensable. And so to test this, one shut down this pathway by starving cells of citrulline or knocking out ASS1. And in both cases, this causes dramatic inability of the cells to maintain arginine homeostasis and arginine levels, and really compromises their ability to proliferate. And this is something we're currently following up on to see if inhibiting this pathway can impact tumor progression in animal models of pancreatic cancer. And we're quite excited that this might be something we could target with a relatively large therapeutic index, because arginine starvation is not something that's likely to be going on in other tissues in the body. All right, so I'll stop there and sum up some key take-home points. And so the first thing that I really want to highlight is cellular metabolism. We've really started to think about this as not just something that's totally cell intrinsic, but this is something that's really a dance between the cell and its environment. And increasingly, we've come to appreciate that these cells are more or less equal. The environment and the cell are more or less equal partners in this dance. The unique nutrient availability in the microenvironment of tumors, we do believe this is one of those key environmental factors that is impacting how tumor metabolism actually functions. And we're quite excited now that we've developed some tools where we can go in and actually measure physiologically what's going on in the microenvironment, that this can give us some insight into the key constraints that these tumors are facing, and also allows us to build new cell culture models that mimic these in vivo stressors and ask how the cells act and function under those constraints. And lastly, one key take home is that the tumor microenvironment really can elicit cellular states and phenotypes and behaviors that you just don't see in standard culture models. And we really like to think about this as a lion out on the Serengeti, out in the wild, might have a bunch of behaviors that it's going to require in order to survive those conditions, both defensive and aggressive types of behaviors to get the nutrients and defend itself. In contrast, a lazy lion in a zoo that's fed a steak every day, which is kind of what we're doing with our cells in culture, might not exhibit a lot of those phenotypes and behaviors. And so we really need to think about that as we think about modeling PDAC cells in vitro. And so with that, I'll stop here and thank everybody in the lab, especially Juan and Lindsay, who did the bulk of the work. They got a lot of help from Riona and Patrick, all of our collaborators at MIT and U Chicago, and of course our funding sources, and especially PanCAN. Thank you very much. We're doing questions.
Yeah. We are doing questions.
Oh,
are we?
sorry. Yeah. I
Yeah.
didn't know if we were doing them now or after.
Yeah, I didn't get instructions otherwise. Is that okay? That was terrific, Dr. Muir. Are there questions from the audience? I have one. There's one.
Hi, I'm from the University of Miami. I was wondering if you did your experiments under anaerobic conditions with the cells,
Oh, under.
or just aerobic conditions?
Oh, sorry. Can you say, I didn't hear the first part. Under which conditions?
So I was wondering if you were able to look at what you were studying under anaerobic conditions, because there is hypoxia in tumors, so, or just aerobic conditions.
Yeah, that's a great question. So I didn't indicate it in the talk, but of course, hypoxia, oxygen is kind of another nutrient in a lot of ways. And so all the experiments, we do them at 0.3 to 0.1% oxygen, because that's, what's been measured in the human PDAC studies.
Thank you.
Mm-hmm.
So I have a question about, well, first of all, I'm struggling with how you squeeze fluid out of a, you know, the
yeah.
tissue. But the thing that I'm struggling with is that I would think it would be very different, the interstitial fluid around the epithelial compartment might be very different from the fluid in the desmoplastic compartment, the non-epithelial compartment. And I just wonder, do you think that's true? And does it matter? And does it affect the results that you might be producing?
Yeah, that's a great question. Kind of a big drawback of what we've done so far is we're basically taking the average across the entire tumor bed and assuming that that's true for every space, like every individual spatial location. And the extent to which that might be true, we really don't know. We've started to do some studies using perfusion markers to mark the poorly perfused from the highly perfused regions and then dissect them away from each other and isolate the interstitial fluid. And so hopefully those kinds of things will start to address your question a bit.
I was just thinking one of the things you might consider, I don't know if you're using primary resection specimens or metastatic specimens, but if the metastatic specimens are a lot more cellular, and so maybe if you looked at more cellular specimens, you could compare with just to see, whether there is a difference.
Yeah, and I think that's a good experiment that we're really hoping to do. Also, just because we want to see, is the primary tumor microenvironment actually similar or different to the metastatic environment in the first place, just to know.
Great. I see one more person coming up.
Nice work. I'm Gloria Su from Columbia University. I'm out of curiosity that if I guess one is that wondering why you use RPMI as the comparison instead of mouse when you're doing the Venn diagram, and whether if you do mouse as the baseline, would you see a different gene overlapping? And the other question is, instead of Venn diagram, if you just look at the profile, do you see it? I was curious which one would be more similar or to mouse, the RPMI or the interstitial fluid, like whether there's any surprises. Thank you.
Yeah, that's a great question. So we can do just like the normal multivariate analysis and blow it up on a PCA map. And so the RPMI cells will be in multivariate space further away from the tumors than the TIFM cells are from the tumors in that kind of analysis too.
Thank you, Dr. Muir. We have to move on.
Thank you.
Dr. Hines is going to introduce the next speaker, but I just wanted to apologize early. My flight was changed out of Fort Lauderdale, so I have to leave out with the speaker. So my apologies. Dr. Hines will stay here though.
Thank you. Our next speaker is Dr. Daria Esterhazy. She's speaking on the intestinal control of pancreatic exocrine immunity.
All right, thank you very much for having me, both to the APA and the PanCAN. And I should disclaim, I appreciate especially being invited as a classically non-exocrine pancreas, let alone pancreatic cancer biologist. I'm an islet biologist and a mucosal immunologist. And to me, all of this is new. So this is my first talk at a pancreatic meeting per se. So I hope I'll convince you that my presence is justified. And so I would like to highlight two hallmarks or some hallmarks of PDAC that really made us enter that field. The first is that early events in PDAC are often preceded, PDAC is often preceded by pancreatitis, which is thought to trigger damage and then repair processes. And then later events probably are that it's an immunosuppressed cancer. So some of the hallmarks of that is that inflammatory dendritic cells and cytotoxic T cells are excluded from the tumor. By contrast, they're Treg enriched, and it's also cancer that suffers from relative neoantigen paucity. So where do we come in? We come in with this gut-pancreas axis, which to me anatomically, or not just to me, clearly also to others, is really a tripartite communication. One is the blood route itself. So the blood that comes from the intestine goes on its way to the portal vein through the pancreas. The second is the ductal route, which we've already heard about in terms of microbial reflux. And the third is the so-called lymphatic route. And I'll talk about two routes. One is the blood route, where both early and late events could conceivably be communicated. One, for example, early on could be that pro-inflammatory bacteria or cytokines could act as triggers, but later also, conversely, immunosuppressive signals from the microbiota or the gut could be conferred to the pancreas through the blood route. By contrast, the lymphatic route could also act early and late, and this is really one of our other major entry points, because it's incidentally such that the head of the pancreas shares its lymph nodes with the duodenum, and they happen to be the most immunosuppressed probably in the entire body. And so these two things are what I'm going to talk about. So let's first go for the blood route. One thing that struck me when I actually entered the mucosal immunology field, which I only did during my postdoc, was that people were talking about these antimicrobial proteins called the Reg proteins in the context of antimicrobial defenses in the gut. Whereas where I had initially encountered them was in the pancreas. And particularly there's one locus called the Reg locus, named such for islet regenerative factor, but its alternative name is pancreatitis associated protein. And so what I did is I just looked at the pancreas whenever we saw actually a fluctuation of one of those proteins that I'm going to show you, Reg3 gamma, in the pancreas and in the gut, and looked at what that looks like in the pancreas itself. So I'll take you through this. Basically what we did is we compared germ-free mice with specific pathogen-free mice. So these are mice that just have the regular lab microbiome over time. So at age of 13 days, post-weaning at 4 weeks, and then 6 to 8 weeks. And what you can see is that in the very beginning, the pancreatic Reg3 gamma is very low. But then as the mice start to be colonized naturally, Reg3 gamma in the pancreas goes up. If you add this very specific bacterium called segmented filamentous bacteria, which notably sits in the ileum, not in the duodenum, you increase this effect, and that persists. However, if you have SFB on its own, and that's a very interesting phenomenon, if you have that monocolonization, with time, the effect wanes, both in the gut and also in the pancreas. So that suggested that there is a communication between the two, and that unlikely is the ductal route, because these bacteria actually sit lower down in the pancreatic, in the intestinal tract. This amount of antimicrobial peptide generation probably in the pancreas is healthy. It probably prevents it from being colonized. However, when you push this a bit further through infections, and that's what we did, and we deliberately chose infections that really target the lower small intestine, again, to not have the confounding effect of leakage through reflux. This might go over the top. So what we did here is we had a form of salmonella or toxoplasma that doesn't do much compared to the non-infected. However, if you go, for example, for Citrobacter, which is an animal model for EHEC, Reg3 gamma was much more elevated. And then if you infect with Yersinia pseudotuberculosis, it actually goes through the roof. And despite our small cohort of pathogens, as it were, a pattern started to emerge. Incidentally, actually, it turned out that all the pathogens that cause this, what's known as an IL-22-Th17 axis, seemed to correlate with this increase in pancreatic Reg3 gamma or potentially pancreatitis-inducing antimicrobials. And so if this is going through the blood route, really there's 3 things that can go into the blood. It's either microbial products, arguably the IL-23 or the IL-22. But you don't necessarily need the bacteria themselves to go to the pancreas in order to elicit this. And so this is ongoing research, but what I'll share is that, for example, upon Yersinia, we see this increased blood level of IL-22 that, of course, would, its first pass organ, as it were, would be the pancreas. So then let me switch to the other side of the coin, and that is adaptive immunity. I mean, this is hardcore innate immunity. This was not even hematopoietic cells. Now we go to full-blown adaptive immunity, and I'll take everybody just briefly through the gut draining lymph nodes of the mouse, which I don't think we've seen so far. So up here, just right under the portal vein, you have the liver draining and the celiac lymph node. And then up here, at the end of sort of the thoracic duct, you have the duodenal lymph node. If you flip the duodenum and then look over here along the ascending colon, you have the ascending colonic lymph node, the jejunal lymph node, the ileal lymph node, the colonic lymph node. These are also known as the mesenteric lymph nodes. So normally the way you assess which lymph node drains an organ is, or a convenient way is to inject dye into the organ and then see which lymph nodes become blue. In practice for the pancreas, that's not very practical because it's such a huge floppy tissue, so that at best you would hit one lymph node. So we took a different approach, and that is we expressed the protein tomato in either the liver or the gut or the pancreas, and then gambled on some of those tomato positive cells dying and being taken up by migratory dendritic cells that then go to the lymph node, and that is how we would recover which lymph node was drained by what organ. So if we do that for the liver, this is what we see. So here we express tomato under the albumin promoter, and then if we look at these CD11C positive cells that are tomato positive, as you may expect, you see the liver draining lymph node and the celiac lymph node, so the two up here light up. If we do this for the gut, now we express tomato under the Willim promoter. You don't see anything in the liver draining lymph node, obviously. Oops, here, let's see. But of course, in all the gut draining lymph nodes, and the inguinal lymph node is a skin draining negative control. If we now do this for the exocrine pancreas by crossing the tomato mouse with PTF1A, we see initially nothing. And that is probably because there's not a lot of cellular turnover. It's almost a post-mitotic tissue. However, when we cross it to an inducible diphtheria toxin receptor mouse and kill some of these exocrine cells, this is what we see. And so you have some, the predominant drainage into the liver and the celiac lymph node, and then the duodenal lymph node. But of note, the celiac lymph node also drains the gut. And so this leaves us essentially with this co-drainage map in the mouse, where we have the liver draining shared by just pancreas and liver, the celiac shared by all three, and the duodenal lymph node shared by liver, by pancreas and duodenum. So now we know where to look for adaptive immunity to the exocrine pancreas. However, we did not have actually an exocrine antigen tracking system. So we generated one. And so what we did is we made 2 mice actually using CRISPR-Cas that express 2 versions of ovalbumin that I'll highlight here. One secreted form of ovalbumin and a cytoplasmic form. And the reason we did both a cytoplasmic and a secreted form was that based on our tomato data, and tomato is cytoplasmic, we were not particularly optimistic that antigen from a cytoplasmic protein would actually be available. And then what we do is we made these mice, cross them to PTF1A, and then now this allows us to have a system where we can track responses to CD4 or CD8 T cells because those already exist in response to ovalbumin. So here I'm showing the data for CD4 T cells that we just adoptively transfer into these mice, and then 3 days later we look in the lymph nodes. So interestingly, and so we were glad that we did this, the effort of making multiple mice, when you look at the proliferation of these CD4 T cells in the mouse that expresses cytosolic ova under the PTF1 promoter, you don't see any proliferation. However, if you do it with a mouse that has secreted ova, you see this beautiful and massive proliferation in the 3 lymph nodes that you would expect. And that's probably akin to a secretory protein from the exocrine pancreas. So with that system, I'll take you through the last and most complicated slide. We took this into the situation where we now know that the duodenal lymph nodes are co-drained. I've told you that they're the most immunosuppressed. However, we already previously found that we can modulate these duodenal lymph nodes and render them more pro-inflammatory by virtue of an intestinal infection that really stays in the intestine, does not spread to the pancreas. And we asked, what happens if we change that lymph node environment in which you see exocrine pancreatic antigen? And so we have our PTF1A as ova mouse. But what we're going to do is we're going to infect it with a reovirus, T1L, that again only infects the intestine. And now transfer in CD8 T cells that recognize ova, knowing that it's the cytotoxic T cells that we need in order to kill a pancreatic cancer, and then ask what happens. So we harvest 3 days later. We could also do it later. The reason we do it 3 days is that the cells have not left the lymph nodes yet. Otherwise, we'd have to co-administer another agent to prevent the lymph node egress. And so here you see the proliferation, as you would expect, of course, in the lymph nodes draining the pancreas, so the liver, the celiac, and the duodenal you see most, and then it wanes over here. And then when we look at the cytotoxicity, very excitingly, and this is just after 3 days, you see this massive expansion of interferon and granzyme B positive cells that recognize exocrine pancreatic antigen in the lymph nodes that they share with the gut. And this is even more extreme if you take all the granzyme B positive cells. There's also some that express TNF alpha and some all three, etc. So you have the whole spectrum. So in sum, what I've told you about is our contribution here is that maybe some of those early pancreatitis events come from the gut, but not through the pancreatic ductal route or microbial reflux, but really through the blood route. And our results seem to suggest that bacteria, or as a matter of fact, any other infection that triggers the IL-22 Th17 axis, this also is true for fungi, for example, may lead to a gut-induced pancreatitis. On the later event or immunosuppressed cancer side, our sort of working hypothesis is that part of the aggressiveness of PDAC might be due to the fact that it happens to unfortunately co-drain into these most immunosuppressed lymph nodes. This might be very beneficial to, for example, suppress type 1 diabetes, but it might be fatal in the context of PDAC. However, that selectively rendering these lymph nodes inflamed may be a strategy to activate the exocrine pancreas reactive cytotoxic T cells, leaving the pancreas itself initially untouched. And so with that, I would like to thank the team and the environment in which I do this. I only joined the university 3 years ago, so a lot of our work was done under COVID conditions. This is my lab, where it's University of Chicago is a great environment. And we have a lot of collaborations and sort of technical support. I would like to thank the funding, not only, but most notably PanCAN, but also the Cancer Research Foundation and some junior investigator grants. And with that, I would like to thank you for your attention, and I'll be happy to take questions.
Questions? First, I'll start off with a question. I mean, really fascinating, and the mouse models are very impressive. So take us through, you presented some preliminary information. Take us through what is the step from inducing the pancreatitis in the models that you're investigating? How can you make that final connection to actually adenocarcinoma? How are you going to make that connection?
To pancreatic cancer?
From the data that you use
yes.
now.
So it took us a while, now we have it. So we have the KPC model, the spontaneous and the aggressive one. And the idea is to see if we colonize them with a certain microbiota, do we accelerate the onset? That's how we're going to do it.
Please.
Okay, great talk, Daria. And I just have a quick question about, have you also looked at pancreatic lymph nodes as well?
Excuse me again?
Pancreatic lymph nodes. There's a lymph nodes inside of the pancreas as well.
Oh, so that's them. So they are heavily embedded in the pancreas. Those are the pancreatic lymph nodes. It's just they happen to be co-shared with everybody else.
So you guys are also trying to look at whether there's any interaction with the mesenteric lymph nodes and then pancreatic lymph nodes?
Yeah, and there isn't any. Yeah, so we looked in each of them and we don't see any reaction to, well, in the tomato system. With the secreted ova system, it's slightly different. In that, obviously, you secrete into the gut lumen. And some of that ova, is not fully degraded. So it actually, some of it is still taken up in more distal lymph nodes. So that might be another way. I don't know how, we'd have to measure, for example, elastase levels or lipase levels in the lumen to see whether that route can also normally happen. But let's say direct drainage of the exocrine pancreas does not happen in the mesenteric lymph nodes. We've excluded that. Yeah. Mm-hmm. So in the mouse, that's all there is. And I think in human, all the surgeons tell me better. There's an addition of the splenic lymph nodes. So there is additional lymph nodes that the pancreas has, but those are in the tail. What's intriguing here is that this is really, this co-drainage happens where most of the PDAC also arises, which is in the head.
I just have one quick question. That was a beautiful presentation, by the way. If I understand it right, you're hypothesizing that selected bacteria promote pancreatic cancer through recurrent episodes of pancreatitis, so creating inflammation over time. How would you approach that, understanding that in a human being?
So very good question. There are these now emerging, these Th17 inducers even in humans. And so what you could do is really in the stool sample, look for those bacteria. There's a whole plethora now that's, it's actually also associated with other diseases like psoriasis, etc. And then screen for those and then see, it was actually Florence McAllister, McAnderson, who, MD Anderson, who contacted me to look at this together and see whether we see that correlation between those bacteria that do that, and then looking at the pancreatitis, potentially even a mouse model in autobiotics. Yeah.
Excellent talk. Thank you very much.
Thank you.
All right, our final speaker is Dr. Krishna Patra. Dr. Patra is speaking on oncogenic circuitries of GNAS mutant pancreatic neoplasms.
Okay, I would like to begin thanking APA and PanCAN for this great opportunity to show our work here. And I would also like to thank you all sticking to the last presentation of this meeting.
It's the best. It's the best. Thank you.
Okay, so this is how I. Okay. This one. Sorry about that. So my lab is interested to understand the genetic mutations associated with pancreatic cancer and how they are involved in pancreatic cancer initiation, progression, and maintenance. So today the talk or the work I'm going to show, it's on a specific mutation which is associated with a cystic subtype of pancreatic cancer. For the interest of time, I will skip the introduction to cystic tumors, which are very nicely covered in a couple of talks in last 3-4 days. But I would walk you through the work we did on this signature mutation, which is found in IPMN subtype of cystic tumor. So to begin, pancreatic cancer is associated or progressed through two different genetic precursor lesions. One is called PANIN, which is very the PANIN, which is very nicely studied in animal models, and another precursor, which is very less studied, is called IPMN, stands that it abbreviates for intraductal papillary mucinous neoplasm. So like its kind of PANIN counterpart, often IPMNs harbor mutation in oncogenic KRAS, but they distinguish themselves from PANIN by having this mutation in the gene called GNS, which is found in 40 to 50% of IPMN associated pancreatic cancer. And later stage of development, these precursor lesions get additional mutation in P53 and other tumor suppressor, and finally they progress to pancreatic ductal adenocarcinoma. So to give a little introduction to GNS, so GNS encode the alpha stimulatory subunit of heterotrimeric gene protein complexes and often studied or mostly studied downstream of G protein coupled receptors. Upon binding of ligands to upon binding of ligands to GPCRs, activate GNS. GNS interacts with adenyl cyclase, generates cyclic AMP, and cyclic AMP working on pKa regulates variety of physiological processes. The Cyclic AMP and PK signaling downstream of GNS is self-limiting, but when GNS is mutated in this particular residue in arginine 201 to C or H, it makes this protein GTPase deficient and makes this it makes it constitutively active. And as I mentioned that IPMNs and associated tumors, we find this mutation in 40 to 50% of the cases. And if you analyze TCGA data, you will find 8% of cases you have this signature or hotspot mutation. And what is unusual with this IPMN that although KRAS is considered as a driver of pancreatic cancer, but in IPMN you will see coexistence of, or in high-grade IPMN you will find coexistence of both the KRAS and GNS. So we were interested to study the mutation or IPMN or GNS mutant IPMN, and we wanted to ask these three important questions. First, if mutant GNS is a driver of IPMN, then what are the mechanisms by which mutant GNS drives cancer, and how this genetic subtype or histological subtype is different from other cancers or other pancreatic subtypes at a molecular level. So when we began our study, there was no model systems available. So we invested a lot of time developing good animal models and human patient-derived system to study the function of mutant GNS in pancreatic cancer. So to what we did, we first placed mutant GNS or this arginine 201C version under the control of a tetracycline promoter, and added the other components of the system and targeted the whole this mutation to mouse pancreas by PTF1-3. And as KRAS is frequently mutated with the GNS, we also combined this animal with mutant KRAS. So we created, we generated GNS only animal, KRAS only, and KRAS GNS. And with the help of our clinicians, when I was in MGH, we could able to, I forgot to mention that this is, as this is a doxycycline inducible system, then we can just add doxycycline any time point of the animal's life cycle, and we can turn on or off GNS or mutant GNS expression whenever we want. Again, with the help of our clinicians, we developed GNS mutant PDXs, organoids, and primary cell lines. And these two complementary system, mouse and human systems, give a kind of really valuable system to understand the mechanism by which mutant GNS drives pancreatic cancer. First, we gave our animals doxycycline, and at 4 weeks of age, all these three genetics models I mentioned in previous slide, and followed the tumor kinetics. As you can see, both KRAS and GNS, when they're expressed alone, they're very weakly oncogenic. But there is a profound synergy between KRAS and GNS when they're co-expressed, and you can see that all of our animals died at 13 weeks of age due to high burden of these IPMN lesions. And when we analyzed through histopathology, we found that in fact our animals make IPMN subtype of lesions, which is very distinct from PanCAN. And although this KRAS and GNS combination made high-grade IPMN, but never progressed to pancreatic ductal adenocarcinoma. Following human genetics, we crossed this animal to P53, and now we could make this IPMNs to progress to pancreatic ductal adenocarcinoma. So thus we established a progression model of GNS mutant IPMN-driven pancreatic cancer model. We took advantage of our doxycycline system, then asked the question whether mutant GNS is required for tumor maintenance. So we generated or developed tumors in those animals, and then when we detected tumor, we took doxycycline off from half of the animals and followed the tumor kinetics. You can appreciate here all those animals, so those are going to be sacrificed within 13 weeks of age. Now they live more than 30 weeks or longer. Eventually, these animals died because they have a constitutive activation of KRAS in the background of GNS, but you can see that but it significantly increased the lifespan. And this is also true in our advanced KRAS GNS PPP3 model. So summarizing what I showed in this section, that GNS is required for IPM initiation, progression, and maintenance. While animal models are great to study to do preclinical studies or the biology in vivo, but it's very difficult to study mechanism. So in order to study mechanism, we generated or established organoid culture system. And as you can see here, that these organoids require continuous expression of a mutant GNS to survive or to grow. When you turn off GNS by removing doxycycline from the culture, these organoids do not grow. And when you implant these organoids in animal model or SCID animal or NSG animal, again, the growth of these tumors are very dependent on mutant GNS expression. So these organoid models actually gave us a tractable system to understand the biology of mutant GNS in pancreatic cancer. Then we have showed that, in fact, mutant GNS activates the canonical PK signaling. And if you inhibit PK by genetic means, you can dramatically reduce the proliferation of this GNS mutant tumors. We went further and wanted to ask what is downstream of PK or GNS that drive GNS mutant tumors. And for this, we did multiplex proteomics, phosphoproteomics, and RNA-seq. And as you can, and summarizing in this cartoon, that what we found that mutant GNS generates cyclic AMP, it activates PK, and it inhibits a family of kinases called 6 salt-inducible kinases that is studied downstream of LKB1 tumor suppressor. And this SYK inhibition by mutant GNS is functionally important, because when we knocked out SYK in the absence of mutant GNS, now we can restore colony formation. But you can see here, appreciate here, that with GNS, you see lots of organoids or colonies, and in the absence you don't see, but in the absence you can knock out 6, you can restore the colony formation, and that we proved both in vitro and in vivo. Then we all know that, I think I have it. Next slide, yes. Then we try to understand what is downstream of 6 that drive tumor. I'm sharing a very one small piece of data here, that what we figured out that, in fact, although 6 have multiple effector, actually CRTC family of co-activators are responsible for the growth of GNS mutant tumor. And interestingly, knocking out CRTC does not affect the growth of GNS wild type tumor. So specifically, this is required for the growth of mutant GNS organoid. Then we also, we know that, although signaling is the engine, but you need a fuel to drive the car. So we analyzed our both proteomics data, and we found an enrichment of fatty acid metabolism, which is that happens in both in the mitochondria and the peroxisome, is highly enriched in GNS activated condition. Again, summarizing everything in one slide, we did multiple functional analysis, and we found out that, in fact, mutant GNS tumors they have a high fatty acid oxidation. And if you inhibit fatty acid oxidation by inhibiting CPT1A, by genetically or with a small molecule inhibitor of this pathway, you can dramatically reduce the growth of GNS mutant tumor, but not GNS wild type tumor, suggesting that mutant GNS driven fatty acid oxidation is important for the growth of these genetic subtype of tumors. So We always wondered that when we were doing our analysis, we are turning on this gene by doxycycline or turning off by removing doxycycline. And in the doxycycline presence of doxycycline, these organoids proliferate. And we are always wondering whether the phenotype we are seeing is just associated with the proliferation. So in order to clarify, we compared the pathway, or we discovered the PK-SIG pathway between GNS wild type and GNS mutant organoid. As you can see, doesn't matter whether it's a low grade or a high grade IPMN, it's always this PK signaling is high, and that and SIG signaling is low. And also through my proteomics analysis, the metabolic pathway found, it clearly distinguishes GNS wild type from GNS mutant tumor. So summarizing what I showed, mutant GNS is required for tumor initiation, progression, and maintenance. SIGs are the potent tumor suppressor downstream of mutant GNS. SIG target CRTC is important for growth. Then PK-SIG signaling and fatty acid oxidation metabolism distinguish GNS mutant from wild type tumor. With this, I would like to acknowledge my lab members, my team, Pancreas team in University of Cincinnati. And most of the work was done when I was with Dr. Nabil Badisi at NGH. So I have a fantastic collaborator, Dr. Liz and Dr. Fernandez. And finally, I would like to thank PanCAN for supporting our lab with a career development grant. And thank you all for your attention, and I'm happy to take any questions.
Excellent talk, Dr. Batra. We have some questions.
Hi, I'm Marta from MD Anderson. I was wondering, how do you explain that the mice that are like KRAS GNAS mutant don't develop PDAC, like even a small percentage of them? Since in humans, we do see that some patients that have KRAS and GNAS mutations do develop PDAC.
Okay, so there are so many other mutations you find in the late stage of tumor. And we, in our animal model, we actually spend a lot of time just asking this question. So what we did, we took that piece of tumor from KRAS genus mouse and tried to transplant again and again and try to grow this tumor for years in SCID mouse, hoping that one day we will get a pancreatic ductal adenocarcinoma from those tumors, but you could not do that. So either, so then we went to the human genetics, then we crossed it, and we found that the P53 is a frequent mutation, and then crossed the animal with P53, and we found the tumor. The question you're asking, there are some KRAS GNS tumors, the PDACs. If I understand correctly, we were asking that they don't have P53 mutations. Is that right? Or?
They don't have P53, but they still develop cancer.
I would suspect they would have some other mutations.
Yeah, we do have the KRAS GNS model in our lab, and ours do develop PDAC. So that's why I'm wondering what the difference is.
Yeah, so that's also another, I'm aware of that animal model you were talking about. So one of the thing we really took care in our animal model, we knew from the very beginning, the level of GNS expression can really make this animal model a messy animal model. So the way we designed animal model, we placed only one copy of mutant GNS, and that we under the control of tetracycline. So most of the transgenic animals, they're not really very, means we don't know where the gene integrates and what the expression level. And also if you look at, you can look at the paper in the supplemental data that we showed that if you take this organoid, in vitro culture condition, you can show very nice GNS expression. But if you implant in vivo, you see extremely very little expression of this mutant. But this is sufficient to drive a tumor. So I think the level of GNS expression is one of the difference between your animal model and our animal model.
Interesting. All right, thank you. Good job.
Hi, great talk. Kyle McAndrews, University of Nebraska Medical Center. So I was wondering, so you use the PTF1A Cree, which is active in the mouse embryo, correct? Have you thought about looking at like a Sockstein Cree ER or PTF1A Cree ER where you activate it in the adult, either ductal or acinar cells?
Yes, we have done that. So again, this is published in the paper that when we generated KRAS P53 model, and we knew that if we activate P53, first of all, these animals develop only KRAS GNS animals, they only live for 13 weeks. If we, on top of we add P53 from embryonic stage, then I don't think we will even get any animal. So for that, we developed a PTF1 Cree ER model, and where we can transiently activate a Cree, and we can do focal lesions, and those animals developed similar tumor, a pancreatic kind of tumor with KRAS GNS, and then when we add P50 on top of it, they progress to pancreatic ductal adenocarcinoma. In short, yes, we have done that.
So is there similar disease between the acinar initiated and the ductal initiated?
So yeah, I did not answer your SOX9 question. Yes, so we have also made animal with, we have done those experiments for SOX9, but what we find that in the SOX9 lineage, the penetrance is not 100%. We don't know because it's whether our RTT is not fully expressed in SOX9 lineage, or SOX9 lineage tumors are very kind of resistant to transformation. But I can share with you that we have done other models targeting exactly the same way with for IPMN specific models, and SOX9 equally gives a tumor to like our PTF1 model. So it's I can, so yeah, means both lineages can give IPMN.
Awesome. Thank you so much.
Well, Dr. Matra, thank you very much. It was our patrons, an excellent talk. And thank you to all the speakers, and congratulations on the excellent work you're doing with PanCAN support. This concludes our 52nd annual meeting of the American Pancreatic Association. I really appreciate everybody staying this afternoon, and downstairs there is lunch if you would like it. Thank you.
Thank you.